Hello students, today we will start with Mycobacterium tuberculosis. Objectives are classification and biochemical characters, antigenic structure, viability characteristics. Now Mycobacterium, it's a slender bacilli and sometimes exhibit filamentous forms resembling fungal mycelium, and so it is named as Mycobacterium. Hansen in 1868 discovered the first member Leprabacillus. Robert Koch in 1882 isolated mammalian tuberculi bacillus, and John in 1895 described M. paratuberculosis, John bacillus. Now, a group of miscellaneous mycobacteria distinct from human or bovine tuberculi of low pathogenicity recovered on occasion from human source. Okay, so genus Mycobacterium, they contain 50 well-defined species, and which include causative agents of tuberculosis, leprosy, chronic hypertrophic enteritis of cattle, and saprophytes. Fine, classification is done on the three things: strict pathogens, atypical mycobacteria, and saprophytic mycobacteria. Strict pathogens, we are looking Mycobacterium tuberculosis. Okay. Next, Mycobacterium tuberculosis and Mycobacterium bovis are typical tubercle bacilli, and both cause human lesions such as pulmonary tuberculosis. And Mycobacterium bovis is excreted in the milk of infected cows. Now let's move ahead with Mycobacterium tuberculosis. Now this genus includes pathogen known to cause serious disease in mammals, including tuberculosis and leprosy. Fine. Mycobacterium are characterized by the possession of very thick, waxy, lipid-rich, hydrophobic cell walls. Being hydrophobic, they tend to grow as fungus, like pellicles on liquid culture media. Hence, the name Mycobacterium is given. They are immobile, slow-growing, rod-shaped, gram-positive bacteria with high genomic G+C content, 61 to 71%. They have a special staining characteristic under the microscope mediated by mycolic acid in the cell wall. They are called as acid-fast staining, Zell-Nelson staining. Okay. In the history, I have told that Robert Koch is the first person who discovered Mycobacterium. Okay. Now, it's a part of a group of bacteria known as NTM, non-tuberculous mycobacteria. Mycobacteria tuberculosis is spread through air. And MAC is a common bacterium found primarily in water and soil. Fine. And this, in addition, whatever is given, M. tuberculosis complex and all, it is for your information. Biochemical characters. These are the different basic characteristics given and their properties for Mycobacterium tuberculosis. Okay. It stains positive for acid-fast stain. Amidase test is positive. Neutral red test is positive. Nicid test is positive. Nitrate reduction test is positive. Peroxidase test is positive. Pyrazinamidase test is positive. And urease test is positive. Okay. Going to antigenic structure. Antigenically, they are homogeneous. They possess two types of antigens: cell wall insoluble polysaccharide antigens and cytoplasmic soluble protein antigens. Group specificity is due to polysaccharides present in the cell wall. Fine. This includes arabinogalactans, lipoarabinomenin, and also lipids, glycolipids and peptidoglycolipids. Fine. This is the structure showing the antigenic variation: cell wall antigens, cytoplasmic antigens. Okay. In the next lecture, we will see its pathogenicity, pathogenesis, treatment, prophylaxis, laboratory diagnosis and symptoms. Fine? If you have any doubts, do ask. Thank you.